everyone, so nice to have you here again. My name is Shaheen, as you all know me, master's student of drug discovery and safety, and today we're going to continue with our lessons about science, evidence, and theory. So let's just quickly recap what we learned last time. We learned that hypotheses are supportable, they are unprovable, and they are refutable. And the same thing holds true for theories, as theories are a bundle of accepted hypotheses, and facts simply explain events. They do not explain why something happens. He said that if we could find a very complex system that was not reducible anymore, and that the parts of this system could not have originated on slowly evolving systems, then his whole theory would fall apart. The question that we had was whether Darwin and his teammates, his scientists in that time, did they really think the cell as a blob of protoplasm? Now, what does a blob of protoplasm mean? It is simply that a cell would be not complex at all. Now, looking through some things that Darwin himself actually published and his colleagues, and as a scientist you have to be critical. Don't take my word for it, don't take their word for it. You have to challenge the theory yourself. Go digging, go search for it. Now, did Darwin have any idea about how complex cells were? Actually, I found a piece about his research on this website. You can go through it and read for yourself. You can just pause it. And here he was actually referring to the things that he found, which were pretty astonishing and amazing about the cells. He was experimenting with tentacles of plant cells, and he was adding chemicals to it, and you could see some things happening in the cell. And he saw these under the microscope. I will share with you what he saw. He saw this picture, and each time that he was adding chemicals, you would see all these spheres coming around. So he and his colleagues saw that the cell was not just something simple. There was a lot of things going on when you add some chemicals, and if you don't add them or add other chemicals, some other things will happen. And it was always repeatable. Every time you add these chemicals, you saw the same results. So they were already getting an idea that cells were much more complex. And this was published 23 years after his publication of his book, so he already had plenty of time to get some new knowledge and insights about how cells are. Now, the complexity of cells as we know them today, we know that the simplest cells are even much more complex than the whole Hubble spaceship, which we have sent into space. So our question would then be, how do these cells become so complex? Now, let's also remember what Darwin said about his theory. You can refute his theory if you find a complex system that you cannot deduce anymore in simpler terms, which could have evolved in individual parts. But there may be some very simple cells that don't have this. For example, in science we have prokaryotes and we have eukaryotes. Prokaryotes are single-celled organisms in which they only need one cell to coexist, and eukaryotes are multi-celled organisms. So perhaps the most simple prokaryote, single-celled organisms, is slightly less complex. So with that, I want to challenge you to find a specific prokaryotic cell or any other cell that is very simple in nature. And the next time we were going to look at the simplest life forms and if they are complex or that they truly are very, very simple organisms, which could have evolved over many, many years. And with that, I would like to thank you, and I'll see you next time.